Good evening, students. Due to some technical issue, I think you are not heard clearly. I think now it's clear one. So again, I will start from starting onwards. Clear? Sorry for the technical issue, students. So how to study? So good evening. So today study step is how to study. So how to study? How to study means so everybody are saying, sir. So everybody are saying study, study, study. So how to study, sir? There in many YouTube channels and school faculties and Neet faculties are etc. etc. In my relations are everything. Everybody are saying study, 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 sir. How to study, sir? How to study? So each one may follow a separate strategy for study. It is my idea. If it is okay, you can follow it. Otherwise, just ignore it. No problem. First one, the text matter you should convert into the image format. The text matter you should convert into the image format. How you can convert? Why we have to convert means, for example, 2 years back, you can 2 years back you may saw any cinema, or 2 years back you may went any any time you may saw so many natural sceneries, or you may regularly you may watch TV serials. Back you may watch TV serials. So now if you are think means just you can remember all things. For example, 2 years back, if you saw one cinema, now you just you think about that cinema means now you can remember that all things. But 2 years back, 2 years back. But 2 years back, you saw 2 years back, you studied so many things. For example, 2 years back, you studied a subject for board exams. Just you imagine it, ma. Just you think it about inheritance and variations. Can you remember all things? One or two maybe remember. Remaining persons? Surely not. Why means that is not our fault. That is a mechanism of our brain. So long that text, text matters will be stored in our brain for a short-term memory. But in the case of colorful things will be stored in our brain for a long-term memory. Long-term memory. Clear? So, for example, one person likes vegetarian means we have to give a vegetarian food. Then only that person will eat very well. If you are supply non-vegetarian to the particular person, he will eat very poorly. Like that, our brains likes the colorful things because it will be stored in a long-term memory. Image format will be stored in the long-term memory. So we have to convert the text matter into the image format. Clear? Image format. What's the meaning of text matter, sir? So in white color paper contains a black color letters, which is called as a text matter. Text matter. Clear? So it will be stored in a short-term memory in our brain. So we have to convert the text matter into the image or colorful things. How, sir? Is it possible, sir? Is it possible through your continuous funny imagination? Is it possible through your continuous funny imagination? However, you can take example is a plant cell. A plant cell, just you can draw in your notebook by using a different colors. By using a different colors, you can draw the cell wall in one color. You can draw the plasma membrane in another color. The next one, you can draw the cellular organelles like mitochondria, nucleus, or etc. etc. You can draw the cellular organelles by using a different colors. So after that, you can label it. After that, you can label it. Surely your brains will easily capture that picture and automatically just you think it, oh, mitochondria, it having the double membrane system. It is a cristae, finger-like apparatus called as a cristae. Then oxysomes will be present in the inner membrane system like that. While you are seeing that picture, clear? So simultaneously, you can recall the functions of the organelles also. Like that, you can apply. That funny imagination is most important one. But that in this during that crash program time is too short. Time is too short to study. So now you cannot apply because now we cannot apply because due to the short duration. But year long program time we can apply it. I am not demotivating you, ma. Listen, I am not demotivating you. The time is not enough. So because you must study so many things. Biology alone having the 39 chapters. Clear. So so funny imagination very fast. It will take a long time for that conversion. Once your practice remains, no issues for the conversion. Fastly you can pick up that the chapters or everything. It will be support to study not only biology in all subjects. Clear. So that's why I'm saying that I will take a special class for you in a regular program time with the permission of our sir. Clear. So we have to convert the text matter into the image format like that. Sir, that structure wise, no problem, sir. If any other text matters, for example, in photosynthesis, so many cycles there, sir. In biomolecules, so many things. What, sir? You can ask this question. You can draw the flow chart by using a different colors, no problem. You can draw the flow charts by using the different colors. Regularly, you have to which feel that cycle you can draw by using a different in your kitchen regularly one. So it is your brain. This way we can convert the text into the image or colorful format. After that, linking is very important one. Clear. So each and every step is very important. So linking means, for example, just now I told cinema is example. So cinema or TV serials, though this one scene is linked with the previous one. Clear. So that linking is continued up to in cinema or so then only we can remember the very scenes like that. You have to apply in your subject also. If you are studying about that plant cell means you must study about the plant cell structures. Simultaneously, you can study the functions of that, functions of the cells. For example, plant anatomy and physiology of plants. In physiology of plants means the first is what are the minerals are important for the plant growth. So after that, how that minerals will be uptaked, how water is uptaked, how it will be transpirated. Clear. So after that, with the help of that one, how it will be produced food. That is called photosynthesis. called as a plant growth regulators. After growth, how it will be fell down. That is called as a plant growth inhibitors. Clear. So after that, after completion of process, how we can modify that one. For example, that vernalization process or etc. etc. So like that, you can link that each chapter you can link with another chapter. So similarly, the topic also you have to link it. For example, if you study about, you have to study about that gene. What are the experiment done by the done with the help of a hybrid, dihybrid? After that one, so what are the exception of some Mendelian inheritance? After that one, you have to study that chromosomal theory or etc. etc. So you have to link that each topic. After that, you have to learn. After ingredients, you can study about that molecular ingredients, which is easy to understand it. After that, you can compare one topic to another topic. Clear. So in this way, you can remember. So clear. So you have to link each one. That is depends upon your idea. So finally, recall it. Recalling is the most important one. Very first class also, I told about this recalling because continuous revision. Nowadays, no problem. Maximum schools are following the revision only compared to teaching because revision gives some more marks. But that revision alone cannot support to get a high marks in competitive exam. In board exam, no problem. But NEET preparation is entirely different from the board exam preparation because here study is important, then revision also most important than study. The both will be important. So recalling means regularly you have to recall it. For example, if I completed that 1 hour or 1.5 hours class, so after 1 hour or 1.5 hours, you have to recall it. Just 10 minutes, that is more than enough. So you have to read it. So what are the topics are today teached? So just to close your eyes, just you think it. If you have any doubt, just you can open your hints notebook and just recall it. It will support to long-term remembrance. Clear. So in this way, you can recall it. So after 1 week or after 10 days, so periodical time, you can fix a periodical time. In periodical time, you have to recall it. So the 3 things are most important. Text convert into the colorful things. After that, link one to another link. A topic should be linked with another one. The final one is the recalling. If you follow this one, surely we will get a high score. High score. High score means how much? Just you aim more than 700. Surely we will get, no problem. Everything is possible in SURIYA Academy. Clear? If you follow some strategies, this is my strategy. Your wish you can follow, or if you already you are following on strategy means no problem. You can follow it. This is my idea. Next is mutation. Yesterday we discussed about so many things. At last we discussed about mutagens. Mutagens means a mutation causing agent. That mutagens are a physical mutagen and chemical mutagen. Comes under the physical mutagens. Many chemicals are comes under the chemical mutagens like mustard gases or etc. That action is that mutagens directly can attack the DNA of nucleus and the changed the character of DNA. Otherwise, that mutagens may attack the cytoplasm, which brings the changes in DNA. In this way, also mutation will be occurs. So this up to this level we discussed. Today we are going to discuss about a mutation. So mutation means a sudden changes occurs in the genetic material. That mutations are broadly classified into the 2 types. One is a chromosomal mutation. Another one is a gene mutation. Chromosomal mutation, also known as chromosomal aberrations. Here, what's the meaning of chromosomal aberration? Means changes in structure of DNA by addition, deletion, or maybe an inversion like this. So anyhow, the changes will be occurs in DNA. That is called as a chromosomal aberration. It will be furtherly subdivided into the 4 types. First one is addition. Sorry, first one is deletion. Next one is inversion, deletion, and translation. Those are the 4 types of chromosomal aberrations. We will discuss one by one. The first one is deletion. First one is deletion. So deletion means a segment of chromosome is lost. Any one segment will be lost due to the mutation. Due to the mutation. This is called as a deletion. That deletion will be subdivided into the 2 types. One is a terminal deletion, another one is a intercalary deletion. Terminal deletion, another one is a intercalary deletion. Just you can see the normal chromosomes, normal segment of chromosome, B, C, D, E, F, G, H. That is the normal segment of chromosome. Just you can see this one. Here A, B, C, D, E, F is there. Here A, B, C, D, E, F is here. GH will be removed, will be deleted. GH will be deleted by the action of mutation. Here occurred in the end of the chromosome segment, which is known as a terminal deletion. Here you can see A, B, C, D, E, F, G, H. A, B, C, D, E, F, G, H should be there. But instead of that one, A, B, listen here, ma, C, D is lost. Because here the removal is occurred in the middle of the segment, which is called as a intercalary. Inter means a between, middle. So the deletion is occurs middle of the segment of chromosome, which is called as a intercalary deletion. Clear? So in deletion causes a Craig-Chat syndrome. The deletion causes a Craig-Chat syndrome, means that infant having the small headed, while that infants are crying, that voice will be a cat like. So that infant cries like a cat. Those are the symptoms of deletion. The next one is a inversion. So inversion means a segment of chromosome, particular segment breaks. After that, it should be rejoined in reverse order, or maybe altered order. I mean randomly. It will be joined randomly or a reverse order, which is known as a inversion. The segment of chromosome breaks and reunites in reverse order or randomly, which is called as a inversion. For example, this is a normal gene. A, B, C, D, E, F, G, H is present. A, B, G, D, E, F, G, H is present. That inversion also 2 types. What are they? Means a pericentric inversion. Another one is a paracentric inversion. One is a pericentric inversion. Pericentric inversion means inverted segment present it. While it reuniting in third position, but in paracentric, not inverted segment, that is called a paracentric one. For example, here A, B, C, D, E, F, G, H is there. Here I wrote in green color, ma, A, B, C, D, E is present as a EDC, inverted one, EDC, and remaining thing is a same, F, G, H. Clear? That is called as a pericentric one. That is called as a pericentric one. Clear? So paracentric means A, B, C, D, the last 4, E, F, G, H is breaks. After that, it is not present in inverted segments. but that position locus is altered, which is called as a paracentric. Paracentric means not included the inverted segment, which is called as a paracentric. That is called as a paracentric. That the inversion of chromosomal aberrations produces a variations in organisms. Produces a variation in organisms. Next one is a duplication. So duplication means segments are repeated or additionally present. It is called as a duplication. It is called as a duplication. So the first one is ABCD. ABCD, EFGH. That is a normal segment. Here ABCD, EFGH at last already when GH segment is present, again the GH is present. That kind of repetition is called as a duplication. Clear, the segment is repeated. The duplication produced the bare eyes in Drosophila. It may ask Neetma, kindly note it. Duplication of chromosomal aberrations produced bare eyes in Drosophila. The last chromosomal aberration is a translocation. Translocation means non-homologous chromosomes can exchange the segments. For example, ABCD, that is it will be present in the first four position, 1, 2, 3, 4. Then 5, 6, 7, 8 is another segments will be presented similarly. Like that, ABCD, EFGH will be present in the second strand of chromosome, second strand of DNA. So that here that exchanges occurs. That EFGH should be present in 5, 6, 7, 8 position of the first strand. But EFGH is present in the complement strand of next position. Complement gene of the next complement strand of the same position. I mean 5, 6, 7, 8. Like that. Here that crossing is there. So non-homologous chromosome exchange its segments. This is called as a translocation. They are transferring its location. Clear. EFGH should be present in the first strand of 5, 6, 7, 8 position. But EFGH is present in the complementary strand of 5, 6, 7, 8 position. Like that, the transferring its location. That is called as a translocation. Translocation. The translocation of chromosomal aberrations produced a cross-shaped pairing. Cross-shaped structure while it pairing. Cross-shaped. Because that ABCD next one is a EFGH. So it will be present in the another strand. While it is a pairing, automatically cross-shaped structure, we can get it clear. So that's all about the chromosomal aberration, chromosomal mutation. The next one is a gene mutation. Gene mutation means what's the meaning of gene? Yesterday I told functional part of chromosome is called as a gene. Gene is a hereditary units which having the blueprints of DNA. That is called as a gene. So chromosome consists of the gene. You can write like a flow chart. First one is the chromosome having the gene. The DNAs are present in the gene. So basic one is a DNA. Clear? DNA is present. Again, I repeat, DNA is present in gene. Gene is present in chromosome. Which one is the base? Which one is present in bottom position? DNA. DNA is made up of a sugars, then phosphate group. The last important one is a nitrogenous bases, purine, pyrimidines like adenine. In case of DNA, adenine, guanine, thymine, and cytosine. So here that gene mutation means changes occurs in the nitrogenous bases of genes. Nitrogenous bases of genes. The changes may occur in the any one of the bases. Clear? Any one of the bases. That is called as a any one of the base or base encoding proteins, which is called as a gene mutation. But in the case of chromosomal mutation, that changes occurs clear. Any segment is lost or inverted position like that. Here, mutation occurs in the specific bases. That is called as a gene mutation. Already explained the terminology. Already explained the terminology by me. For example, mutant means a organism carrying gene, that is called as a mutant. Mutagenesis means the process of mutation, that is called as a mutagenesis. Mutation means a sudden changes. Clear? So yesterday I explained the terminology, important terminology in mutation. The next one is the types. What are the types of mutation means? So many types of mutations will be there. Many types of mutations are available. So among that one, so few most important one to our Neet. That thing only we are going to discuss is first one is the spontaneous mutation. What's the meaning of spontaneous? Spontaneous means naturally. Spontaneous means naturally. So that mutations occurs naturally. Mutations occurs naturally due to the random interactions with environment. For example, any organisms. For example, in human being, in a daily wages, they are doing their work regularly. In specifically that things are occurs in agriculture field. Specifically, they are doing work under the sunlight, which having the ultraviolet rays like that. So that reactions occurs with the environment. At the time also mutation will be occurs. It is not an induced one, that naturally happen things. Sometimes, for example, if you are taking anything, it may having the some chemicals. So at the time also mutations will be formed. So likewise, that spontaneous mutations are naturally occurring process. It will be occurred due to the random interaction with the environment. Clear? Which is called as a spontaneous mutation. The next one is a induced mutation. So induced mutations means what is the meaning of induced? Already I told in biology, one thing is present, another one should be a opposite one. So spontaneous means a natural one. Induced means the opposite to spontaneous, means induced. So induced by the some mutagens, which is called as a induced mutation. Clear? So that mutation is caused through the mutagen, either physical or maybe a chemical, which is called as a induced mutation. So induced mutations will be caused either physical mutagens or a chemical mutagens, which is called induced mutation. Clear? Induced mutation, yesterday class I clearly explained what are the agents comes under the physical and chemical mutagens. Mode of action also clearly we discussed in yesterday class. Clear? I think no need to explain again. The next one is point mutation. What's the meaning of point? You know very well about them. What is the definition of mutation? Point means a specific one, specific place, specific. So point mutation means that mutation will be occurs in the specific or a single base pair. Specific or a single base pair, which is called a point mutation. So here the single base pair is altered through the effect of mutagen, which is called point mutation. Due to the point mutation, structural changes will be occurs in the single locus. Locus means a position of gene in chromosome or position of DNA in gene, which is called as a locus. Locus means a location. So due to when the single basis is altered means changed or addition, deletion or anything changed. So at that time, what will happen? Automatically the structures will be changed. Structures will be changed in the specific, I mean the single locus. Clear? How the changes is occurred? how that changes occur means how the single base pair is altered means maybe substitution or inversion or maybe a deletion or addition or any other things. So any other things, substitution, we will discuss one by one. The first one is a substitution. So substitution means this is a normal gene. This is a normal gene. So bases you know very well, ATGA, CTG. Because the 4 bases are present in DNA: adenine, guanine, thymine, and cytosine. Remaining things you know very well. Adenine paired with thymine, that thing you know very well. So this is a normal gene. Here, base substitution. Base substitution. In base substitution means, listen, 1, 2, 3, fourth. Fourth base. Here, fourth base is what's the fourth base? Base A. Here A is substituted with T. I mean thiamine. Clear? Thiamine. You know very well meaning of substitution in kabaddi or cricket or anything. The substitution will be there in case that person is suffering from any injury or any other thing. So at that time, that substitute players will be substituted to play the particular game. Like that, in any cases, that specific bases will be altered, substituted with any other one. Clear? So here A is altered with thiamine. Clear? This is called as a base substitution. Base means you know very well. Yesterday also I told adenine, thiamine, guanine, cytosine. In case of RNA, the thymine is replaced by the uracil. That 4 individual is called as the bases. Clear? The next one is a base insertion. Base insertion. Insertion means what is the meaning of insertion? Insertion means we can insert. We can insert one extra bases. For example, normal gene. For example, normal gene. Totally 7 bases are there. A, adenine, thymine, guanine, adenine, cytosine, thymine, and guanine. But in base insertion, adenine, thymine, guanine, again one guanine will be inserted between the guanine and adenosine. So that inserted base I wrote N here. This is called as a base insertion. That is called as a base insertion. Clear? So due to the base insertion also makes a structural changes in DNA. Makes a structural changes in DNA. The last one is a base deletion. Base deletion. Deletion means adenine, thymine, guanine will be there. So after the fourth base, fourth base, here adenosine A is present, but here there is no adenosine. There is no adenosine. So adenine, thiamine, guanine is present. After guanine, again cytosine and thymine and guanine will be presented. Here adenine is deleted. Adenine is deleted. This is called as a base deletion. Those are the example of a point mutation. Clear? So point mutation means a single base pair will be altered. Base pair or base will be altered. Example, base substitution, base inversion, and base deletion. Here the single base is only altered. That is called as a point mutation. The next type of mutation is a frame shift mutation. Frame shift mutation. What is the meaning of frame? Frame means framing the particular thing, which is responsible for the particular one. That is called as a frame. Framed one, fixed one, framed one, fixed one. For example, GGT, GT, G component GT is one triplet code which is responsible for making a glycine means, which is responsible for making a glycine amino acid, because the codons are responsible for that amino acids are responsible for making a protein nonce. So GGT is framed that codon, that triplet codon responsible for the production of a glycine amino acid, like that. It will be framed, fixed. That is called as a frame. That framing is shift. Shift means a changing, exchange, or maybe a changed due to the addition of some things. That is called as a frame shift mutation. So addition or maybe a deletion. So anyhow, that frame should be changed. The triplet codon should be changed due to the addition or deletion of some bases in the triplet codon, which is called frame shift mutation. So inserts or deletes a single bases. Insert or deletes a single bases. Results changes in reading frame. Results changes in reading frame. I will give definition for frame shift mutation. Chemical causes a loss or addition of one or more nitrogenous bases in structure of DNA, leading to complete change in reading of genetics code. So just now I told any one bases will be inserted or maybe deleted in the specific triplet codes. So at that time what will happen? That changes, structural changes is occurs in the triplet code. The changes is occurs in the triplet codes. So while you are reading that one genetic code, so automatically it changes is occurs. Clear? The first one is there is a normal one. You can see the diagram. First one is a normal one. TAC, AAA, TC, G, and GG, and the last one is a ATT. The each triplet codes are responsible for making the specific amino acids. That specific amino acids can make the specific proteins. That thing, univeral tyrosine, glutamic acid, glycine, leucine, like that. Clear? So the second one, listen here. Here that GAA is present, that in a normal one. When that frame shift mutation occurs, at that times it becomes GA and GA. Clear? Here the single base only inserted. In normal, GAA is present. Here GA, after GA, additionally G will be added, inserted between the A and A. Additionally, A will be inserted, guanine will be inserted between the adenine and it gives some changes in the triplet codes. So initial triplet code is a GAA, but after the insertion of guanine in between that A and A, it becomes GA. Clear? So the changes in reading frame. The changes is reading frame due to the insertion of single base per GC, which is called as a frame shift mutation. Frame shift mutation. So just now I told. So automatically, what will happen? It leads the changes in amino acids in structure of proteins. Automatically changes is occurs in the protein structure of amino acid, because amino acids are building blocks of proteins. So one gene is altered means automatically it shows us some structural changes. In amino acids of proteins. So the new protein will be formed. Which is completely different from the previous protein. For example, just now I told here the GLU, glutamic acid, GAA. which is that is a triplet codes. That is a triplet code. It codes glutamine amino acid. That amino acids can able to make the specific protein. That's here that structural changes is occurs in the triplet codes of glutamine or glutamic acid. So automatically that protein formation also, that the protein formation also entirely completely different from the previous protein. Normal glutamic acid can able to produce the particular protein, but after the structural changes, it cannot able to produce the previous protein on original protein on it will be produced another one protein on. So in this way it will affect the protein production. So new protein will be produced. So this is called as a frame shift mutation. So that's all about the mutation. That's all about the mutations. Does we have any benefits from mutation? Otherwise, if we have any dangerous or harmful effect from mutation, what's your answer? The mutation may have or they produce three kinds of effect. What are they means? First one means some mutations may cause a very dangerous, some mutations cause some harmful effects. Many diseases is caused. For example, that sickle cell anemia or so many genetic disorders will be caused by the mutations. Clear, caused by the mutations. Similarly, some mutations occurs in the plants. So at that time, we will get some beneficial effect. We can get a more number of fruits like that. Yield may be increasing because that due to the mutation compared to the normal plant yield, mutated plants may give a more quantities of fruits or vegetables due to the beneficial mutation. Clear. So mutations will give a beneficial effect, both beneficial effect and harmful effects. Clear. Some mutations are cannot produce a beneficial effect. Same time, it cannot produce the harmful effect. Clear. For example, six fingers. Also, regularly we are having that 5 fingers in our hand. Commonly, we are having the 5 fingers in hand. In some peoples may have an extra small finger, little finger. Small little finger is there. And sixth one. So in sixth one finger, we don't have anything. We don't have a beneficial effect. Same time, we don't have any harmful effects like that. May cause like a mole, etc., etc. We don't have beneficial effect. Similarly, it won't cause harmful effects. Clear. So mutation is over. Next one is genetic diseases. Genetic disease or genetic disorders. So genetic disorders means mental disorders or chromosomal disorders. Genetic disorders, the disease. Genetic, that disease occurs. Disorders, it is not in order, called disease. So the disease occurs in gene level. Gene level, which is called as a genetic disorders. Clear. So one is a Mendelian disorders, another one is a chromosomal disorders. So genetic disorders are two types. One is a Mendelian disorder, another one is a chromosomal disorder. So first we will discuss about a chromosomal disorder. Chromosomal disorder. So chromosomal disorder means that chromosomal disorders are caused by the absence or excess abnormal arrangement of one or more chromosomes. If you are taking one chromosome, means for example, human normally having the 46 chromosome means one chromosome may be lost. It becomes a 45 in number. Similarly, we have a 46 chromosome means one chromosome may be added. So at that time it becomes a 47 chromosomes. Likewise, one chromosome may be absent or one chromosome may be exists and arranged in an abnormal one, which is called as a chromosomal disorder. So caused absence or exists abnormal arrangement of one or more chromosome, which is called as a chromosomal disorder. Because that chromosome numbers is a constant. That chromosome numbers will be vary in each and every living organisms. The chromosomes, the human having the particular constant chromosome number, in Drosophila having the particular constant chromosome number, likewise mouse having the particular chromosome number. So that chromosome number will be vary from the each and every organism. In case any changes occurs in chromosome number, not only the chromosome segment and not only the DNA base pairs, in chromosome numbers also should be constant. If changes is occurs, automatically it causes the disease because it cannot able to do the normal functions. Clear? That chromosome disorders are many types. First one is a pleidy. Pleidy means a pleidy. Pleidy means a numbers in chromosomes. The changes occurs in the number of chromosomes. The changes occurs in the number of chromosomes. How it can get changes means loss or gain of chromosome. How it changes its structure. Once it have to loss or maybe a gain. For example, one chromosome is additional or maybe a deletion of chromosome numbers. Pleidy. That pleidies are two types. One is a aneuploidy, another one is a euploidy. Aneuploidy, second one is a euploidy. First one, aneuploidy. Same thing. Now, just now I told what's the meaning of pleidy. Aneuploidy means a gain or loss of one or more chromosomes. Aneuploidy means a gain, addition or deletion of one, sometimes one or sometimes many chromosomes, which is called as aneuploidy, which is caused by the non-disjunction. Aneuploidy, it may ask in NEET exam, aneuploidy caused by the non-disjunction. What's the meaning of disjunction? Non-disjunction. Failure of separation of homologous chromosome during the gamete formation. So that some chromosomes are failure to separate during the gamete formation, which is called as a non-disjunction. Failure of separation of homologous chromosome. That is also important. Kindly note it. That aneuploidies are three types. Aneuploidies are three types. What are they means? Monosomy, nullisomy, trisomy. Monosomy, nullisomy, and trisomy. The first one is a monosomy. First one is a monosomy. Mono. What is the meaning of mono? Mono means a single. Mono means a single. Mono means a single. Mono means single. Somy means universal chromosome. Somes means a chromosome. Mono means a single. Somes means a chromosome. So single set of chromosome. The monosomy means a single set of chromosome. One chromosome is lost from the pair. That is called as a monosomy. One chromosome is lost from the pair. For example, 2n. So you know very well, chromosomes are diploid in stage 2n in nature. So 2n minus 1. 2n minus 1. That in the number of chromosome, one chromosome is a total number of chromosome. Among the total number of chromosome, one will be lost. That is called as a monosomy. Mono means one. Somy means a chromosome. So one chromosome will be lost from the pair. That is called as a monosomy. For example, in Drosophila, totally having the 8 chromosome, one chromosome will be lost. One chromosome will be lost due to the mutation. Clear? So at that time, end of the results, it get a totally 7 chromosome. Totally 7 chromosome. So this is example of a monosomy. In humans also. So monosomic man. Monosomic man means we have only the 45 chromosome. Out of 46, one will be lost. So as a result, 45 chromosome. This is example. One chromosome is lost from the 46 chromosome. This is called as a monosomy. Monosomy. Clear. Women's also. In women's also, the monosomy occurs when the egg is without X chromosome, fuses with the sperm containing X chromosome. When egg without X chromosome will fuse with the next one is nullisomy. Nullisomy means nullisomy. Nullisomy means both chromosome of lost. Nulli, nil. Nil means what? Nothing. Nil means nothing. Somi means chromosome. Nullisomy, nilsomy. Clear. So nullisomy means that both chromosome of pair are lost. The both will be lost. That is called as a nullisomy. The third one is a trisomy. Tri means one chromosome is added. Tri means 3. So already diploid in nature. Pair. Pair means 2. Chromosome is called as a ploidy. So one chromosome is added in a pair. That is called as a trisomy. One chromosome is added in the pair is called as a trisomy. The trisomy is also caused by the disjunction. Also caused by the disjunction. For example, X chromosome will be added to the man. We can get it 46 plus 1, 47 chromosomes. So already we have a 22 AA plus XY. 22 AA plus XY. So additionally one chromosome will be added to the man. So because man only having the human male only having the XY, male only one X chromosomes will be added to the male. So at that time it is a trisomy. At that time the diploid becomes a trisomy. Clear? So XXY. So which is an example of a trisomy. Clear? It is an example of a trisomy. For example, Klinefelter syndrome. We'll discuss later. So that's all about the aneuploidy. The next one is a euploidy. Next one is a euploidy. Euploidy means changes occurs in chromosome number. Changes occurs in a chromosome number. For example, haploidy, polyploidy, haploidy and polyploidy. Haploidy means the gametes carry haploid number of chromosomes. The gametes can carry the haploid number of chromosomes. I mean N number. Haploid means opposite to diploid. Single chromosomes. That is called as a haploid. Gametes carry only the single number of chromosomes, N number of chromosomes during the fertilization. So at that time, what will happen? So that male brings the haploid number of chromosome, female brings the haploid number of chromosome. Fertilization will be occurs on that haploid plus haploid both will be fused and forms a diploidic in nature after fertilization. Clear? So N plus N equal to 2N. So haploid means nothing. The gametes carry only the haploid number of chromosome. So it will, which is undergoes a fertilization process. So during that time, gametes, haploid gametes will be formed and forms a 2n in nature. Clear? The next one is a polyploidy. Poly means a many. Poly means a many. Organisms carrying the more than 2 set of chromosomes. Organisms carries the more than 2 set of chromosomes. Is called as a polysomy. Is called as a polyploidy. Carries that more than 2 set of chromosomes called as a called as a polyploidy. Polyploidy means more than 2 set. Triploidy. It having the 2n plus 1. 2n plus 1n, which is called as a triploidy. Tetraploidy means 2n plus 2n, which is called as a tetraploidy. It will be moves so on. The next one is a pentaploidy. The pentaploids. Pentaploids means 2n plus 2n plus n. That is called as a pentaploidy. The next one is a hexaploidy. Hexaploidy means 2n plus 2n plus 2n, like that. So organism carrying the more than 2 set of ploidy is called as a polyploidy. So that's all about the euploidy. So in human beings, due to the disjunctions produces the 3 types of syndromes, namely Klinefelter syndromes, Turner syndromes, and Down syndrome. Down syndrome. Think time is up, ma. So that non-disjunction syndromes and the inborn error diseases genetic disorders. I mean that Mendelian disorders we will discuss and tomorrow. Why I am saying that topics to be taken tomorrow topics. Why I am telling means just you have some ideas. You have to collect some ideas. What tomorrow we are going to discuss it. That interest makes to study automatically, it will be converted into the marks. That's why I am telling. So what are the things we discussed more today? The first one is a mutation, the types of mutation. The next one is a genetic diseases. We discussed in genetic diseases, we discussed about the chromosomal disorders. Chromosomal disorders not yet completed. Thank you, students. Thank you. You can give your valuable comments here. Thank you. Thank you very much.